Nelms, who's from University
This meeting is being recorded.
again. So I would like to introduce Brett Nelms, who's from University of Georgia. He's at the Department of Plant Biology. And he's a relatively new faculty member there. He arrived August 2020. Or getting there, or there's a mixture of both. So the 5 years before, you were in the lab of Virginia Walbert, Stanford. And that's an area where there's some shared interest, I would say.
Yep.
Around meiosis, could you manipulate it? And he did outstanding work, got 2 publications in Science, among others. So it's really high-profile research that he's doing. Before going to Stanford, he got his PhD at Harvard, biophysics, if I said that correctly. And you mentioned you started out in human medical.
Yeah, I was doing mammalian cell biology in grad school.
And so he got his BS in Wisconsin, and that was all the natural sciences I saw, like chemistry, physics, biochemistry. So it's a broad education, and I think it's helping him to deal with this complex topic. So welcome.
Cool. Thanks a lot. Yeah, so I'm going to talk to you a little bit about a project that began at the end of my postdoc and kind of has continued in my own lab, along with some of the directions of where we're thinking to go with it. So this very abstract piece of microscopy art here, this is a dissecting fluorescent microscope close-up of maize silks with pollen on them. So this kind of dark shadow you see here, these are 3 maize silks, and you can just see the huge amount of pollen that's landed there. And the main thing I want you to take away is look at is how many pollen grains can be competing together on each of these individual silks. So my main title of my talk, The Forgotten Generation, is the haploid plant gametophyte. And a lot of us learned about this thinking about mosses and some organisms where the haploid phase is actually quite visibly apparent and dominant. So here we have moss growing over a fallen redwood tree in Big Basin State Park of California, which is about an hour south of where I was for my postdoc. And huge amount of moss here, and most of that is actually the haploid stage of the life cycle. But if you look closely, you can see these funky little structures coming out, and that's the sporophyte that's most analogous to all the plants that we spend most of our time thinking about. Each of these life cycle stages, it's different ploidy, but they have entirely different reproductive mechanisms. They have different developmental forms. This sporophyte here is asexual. It makes a spore, followed by the sexual gametophyte phase. And it seems weird at times, but this is still true for flowering plants and a lot of the organisms we look at. So here's a close-up of a maize male flower where you can see the anthers, and here's all your diploid sporophyte. But each of these pollen grains here are actually that gametophyte phase. Each pollen grain in maize at pollen shed is tricellular with 2 different specialized cell types, its own gene expression. There really are truly multicellular organisms. And so I'm very interested in understanding what are some of the consequences of this in plants and what can we do with it. So this is a really old micrograph of a close-up of a single rice anther and some pollen grains from a sporophyte plant that's heterozygous for a waxy mutation. So in the plant, you make amylose starch normally when it's heterozygous. This is a recessive mutation, but if you look at the pollen grains from this one plant, and this is stained with iodine, you can see that half of these pollen grains have this mutation, don't make the amylose, and are unstained or weakly stained compared to the other half right there. And that's really, I think, one of the most significant differences between plants and animals here. Here we have the haploid phase segregating phenotypes where you have that link from genotype to phenotype, and we see this for tons and tons of genes in plants. You would never see an image like this with a mammalian sperm or egg cell. Now, there's a huge capacity for selection in pollen, and it's a couple different things. One is it's good to realize that what happens in pollen does not necessarily stay in pollen. For instance, this waxy mutation here, this is actually one of the big differences between sticky rice and non-sticky rice. So it has agronomically relevant phenotypes in addition to phenotypes we can see in pollen. And I mean, even in the 80s, using techniques that I barely understand, but going into the genomic era, one thing people have realized is a lot, if you look at, say, transcripts, a huge portion of transcripts you can detect in the haploid phase, you also detect in the diploid phase. This is an early microarray paper in Arabidopsis, where they found that actually 2/3 of the genome that they could detect in any tissue was found in both the haploid and the diploid phase. So we're really not just talking about those rare pollen-specific genes. Almost everything you can think about with core cell metabolism, cell biology, all of this is active in pollen, just like it is in all of the other cells of the plant. Another thing is that traits that are recessive in the sporophyte can face selection directly in the gametophyte. And you see that immediately here, where this heterozygous plant that would have the dominant big WX waxy phenotype is immediately segregating. And the population sizes are huge. With a lot of the older maize inbreds that I use a lot, you have about 10 million pollen grains per plant. A tree can have, 4 trillion pollen grains. And so effectively, each maize plant is doing more genetic experiments than, a large breeding company is in a year. And just to give you a sense on kind of the scope of what this might do, I wanted to share with you a pretty cool old hypothesis for what actually led to the origin of angiosperms, and that it might actually have a lot to do with pollen competition. So, long ago, botanists have noticed of just how complex and diverse angiosperms are. Their morphological diversity, the ability to be in so many different niches. Angiosperms have been enormously successful. And even when you get to the molecular age, that diversity is paralleled when we look at mutations. Angiosperms have about an 8-fold higher mutation rate compared to gymnosperms. And one idea is that floral structure is actually partly optimized, is a structure designed to maximize pollen competition. And to kind of explain how this works, if you look at pollen and gymnosperms, I couldn't find what species this is, but here's one example of a close-up gymnosperm. You have the egg and the ovule here, and the pollen grain landing close to that. Compare this to Arabidopsis, which is a relatively small plant. The pollen's landing up here and has to grow this long way down to these ovules. And this is the big difference. In gymnosperms, you do not have a closed carpel. And so the pollen grains can land right next to the ovule. So this big track right here, that's an angiosperm innovation in the flower. To show you this for scale, this is what that gymnosperm would look like next to Arabidopsis. And this is generally true. If you look at the length that the pollen tube grows, it's consistently drastically longer in angiosperms. Arabidopsis has longer pollen tube lengths than the longest gymnosperm recorded. And not only this, but the pollen tubes grow a lot faster in angiosperms. Typical rates in gymnosperms, you have about 1 to 20 microns per hour, and it can take months to years for the pollen tube to reach the ovule. Angiosperms, you can go, upwards of 2 millimeters an hour or more, like in the case of maize. So this graph here is a little hard to read the bottom, but this maxes out in gymnosperms at 20, and 20 is this beginning here with angiosperms. The pollen tubes grow a lot faster. And if you look at some early diverging lineages, there were a lot of changes to the pollen tube wall that actually predate the evolution of the flower. So the idea is what this does, first of all, you have a closed track so that nobody can cheat and come in from the side. Everybody's got to start at the beginning of the racetrack. You have to grow much longer and further distances, increasing competition. And to cap that all off, a lot of angiosperms are insect pollinated. And this would be like the gun at the beginning of a racetrack, setting everybody off. That insect could deliver a big burst of pollen all at once. And so really the structure of the flower is sort of the anatomical equivalent of an Olympic racetrack here. So the hypothesis is that because of how much competition can occur in the flower between pollen grains, this is essentially a way to screen out negative mutations really cheaply. Most mutations will break things. Huge portion of the genome is expressed in pollen. And so when you break core metabolic processes or anything that slows stuff down, you can screen those out, allowing for higher mutation rates without having the cost, and that this maybe helped contribute to a lot of the success of angiosperms. So the big thing I want you to take away here is that even though a lot of what we think about in pollen, it's very small, it's often not the stage of the plant that a lot of us care the most about, but it can really have big impacts in terms of what happens through evolution, and I think what we can do in terms of breeding. So I'm going to share two stories today. The longer one will be work that I began with Jenny Walbott in my postdoc. And the question was really, when do these haploid pollen grains begin expressing their own genomes? So, you know, we think about pollen a lot in terms of pollen shed, but there's actually quite a long period of haploid development before pollen's been shed in that flower. You know, for maize, I'd plant a seed in the ground. About 5 days later, I might see the seedling emerge. And when the plant's about this high, maybe 40 days later, you're already done meiosis, and that haploid gametophyte is developing long before you even see the tassel emerge at the top of the plant. And so for maize, for instance, the endomeiosis, the pollen shed, it's about 20 days. So actually the majority of length of time of haploid development is during this phase. And most of what we know for gametophyte phenotypes, things like this, are way over here. So the question was really, when does kind of pollen come of age and start expressing its own phenotypes? When do you express that genome? And you could really imagine a lot of different models. It could be really quickly. Meiosis ends, everything gets decayed, haploid genome's active, and you're very immediately taking on phenotypes linked to that haploid genotype. But you could imagine all sorts of delays that would then shelter those transcripts from the kind of, or those genes from the kind of selection that we see in the haploid phase. And there have been debates about the timing of this for a long time without a lot of great data. There's, you know, even, you know, arguments back when people were first studying the cell wall, when all of the cytologists would say, hey, these components of the pollen coat are made inside the microspore. I can stain these enzymes. I can see they're physically in the microspore. And the geneticists would say, hey, wait, no, we've never seen these segregating in a tetrad here. It's got to be sporophytic. And it took a while to someone realize, well, maybe they're just made from transcripts or proteins that were synthesized before meiosis was over. You know, and so I really wanted to look at this a lot more comprehensively. And coming in, my full expectation was you'll have some pathways that do this, some pathways that do this. So you'll see when I actually got the data back, it's a lot more uniform than I was expecting. And you know, when we think about the plant life cycle, where we have the sporophyte and gametophyte phase, a lot of this project was motivated by what we know, particularly in animals, but also some in plants, about what happens early in the zygote. Typically, the majority of the fertilized zygote products are from mom. And that can have genetic consequences where things in the very beginning follow mom, and that can vary by species. And a lot of mammals, it's within a cell or two that the zygotic genome's activated and you're shifting from parent to offspring. But there are many organisms where you can be hundreds of cells till the zygotic genome starts to take over. And so my question was, you know, what about a similar transition that occurs on the other stages of the life cycle? And with what time and what mechanisms do you shift from parent to offspring? So to distinguish this, I actually used an approach that's really similar to this classic 1920s paper, which is just if you can identify anything in the individual pollen grains and distinguish alleles, you can follow what's happening. And so I used single cell or single gametophyte RNA-seq, isolating individual pollen cells or pollen grains, pollen precursors, and then used RNA sequencing with an F1 hybrid maize plant that was a cross between A188 and B73. And this way, looking at the individual pollen precursors, I could see if transcripts are coming from a mix of both alleles, and therefore they must have been synthesized before meiosis was over, or if they're coming from one allele. And this way you can track on a gene by gene basis across the genome what's going on. To isolate cells, it's actually pretty low tech, and I had previously used this in my postdoc looking at early meiotic gene expression, but essentially these figures are with protoplasts from my prior work, but it shows the technique kind of well. Essentially, I'll put these on a microscope slide with a low NA objective, so a cheap objective, which means I have a big working distance, and it's just a blunt syringe needle. and can pick up the individual cells and place those on a cap of a PCR tube to make RNA-seq libraries. So no specialized equipment. This is a pretty non-standard way for single-cell RNA sequencing, but the thing is, pollen and the precursors are pretty big, which helps make this a lot easier. And the big advantage is I can take one anther, isolate several cells from this, and then either use that anther or the two anthers that were in a developmental unit to stage. So I actually know what stage I'm at with all of my collected samples. So this just was really helpful at making it possible to miniaturize, where I could have much more dense time courses than if I had to pull a bunch of material together prior to cell isolation. So to kind of show you this works, first I'll show you some graphs from before meiosis and mature pollen. So this top histogram up here, this is a single pollen mother cell, or PMC. So this is a diploid cell just before the start of meiosis. And I'm just showing all genes where I could find at least 10 transcripts where I could distinguish one allele from the other. And the x-axis is just the percentage of transcripts that are from B73. So 0 means it's all A188, 100 means it's all B73, or it could be a 50-50 mix. And like you might expect for a diploid cell, there's some spread, but most of it's centered around 50-50. Now, if I look in a single pollen grain, all of the transcripts are coming from one allele or the other. And again, you couldn't really do this in pool pollen because you have half the pollen grains getting one allele, half getting the other. But because the technology's come to a place where it can be miniaturized and we can look at individual pollen grains and precursors, we can really get at this question. Now, there are a lot of reasons why you can see monoallelic expression, right? Parental imprinting, there can be differences in promoter usage, presence-absence variant, and even technical things, like maybe the B73 genome is a little bit better sequenced, so things can map more easily there. But it's really easy to distinguish haploid-derived expression from all of these others because you get correlation along the length of the chromosome. This is a F1 hybrid, so all of these precursors have only had one meiosis to integrate those. And here I'm showing the same thing that was on the x-axis. So this is the single pollen grain, first 3 chromosomes. And if you look along the length of the chromosome, you can see that every gene is coming entirely from A188 for a while till it shifts to B73, shifts back. And so you can directly see the recombination breakpoints from that. And the distribution of the recombination sites on the length of the chromosome, the numbers all fit what we know about recombination frequencies and distributions in maize. So with this working, what I did is I took a time course from just before meiosis all the way through pollen shed. So it's about 28 days in total, with the haploid phase being the last 20 of those. And here's just a heat map of gene expression. It was total about 350 single cells or pollen grains or pollen precursors. And I'll mostly be using single precursors through this talk, just because this includes both bicellular, unicellular stages. But you can see just in gene expression, I have this rich time course. There's some times where a lot of genes are changing all at once. So it's a pretty useful resource just for gene expression. But from here on, I'll focus mostly on that question about when is the haploid genome being activated. So to look at that, one thing I can do is just lay out all the different stages and we can see what's happening. So these stages are much more subdivided. Realizing I wish I put an overall thing, but here we have different stages of meiotic prophase. We have the meiotic divisions. This is later stages leading up to pollen. So haploid starts around here. If I look at all the different cells, I can draw a figure like this for each individual cell, but so that we can summarize across many, what I've just done is arbitrarily decided to label anything between 20 and 80% as biallelic and anything more extreme than that as monoallelic. That threshold's a bit arbitrary, but the results are the same no matter where I cut it. In this way, we can kind of interpret this. So here, the cells before meiosis, you can see they're mostly biallelic, single pollen grains. I'm seeing most expression is coming from one allele or the other. And if I look throughout meiosis, it's kind of a similar thing. Most is biallelic, some of these are falling outside this range, and that's both can be a combination of imprinting, presence, absence, and some of it's just noise. Some of these genes aren't consistently from one allele, but just in noisy count data, you're not going to always fall in the middle. And then when I look at the haploid stage, it was really pretty striking because the earlier stages I was collecting looked pretty similar to what I was seeing before meiosis was over here. And the box plots is a little bit harder to think about how many cells are contributing to this stage here. So I'll show you a histogram on the side. This is just showing, this is all cells before Pollen Mitosis I. So this is going from this UM stage, which is unicellular, to this BM stage, which is bicellular. And here's a histogram of all of those cells. And in blue, I have all of the gametophyte stages after Pollen Mitosis I. And it's this really drastic shift with that one stage boundary. You can see this even looking at the individual precursors. Here's one example of a late microspore, and this is 10 days after the end of meiosis. And most of the transcripts are coming from both alleles. I cannot tell you the haplotype of this precursor here. It's even trying more careful statistics. It is so uniform that I could not figure out, even using chromosome location, what allele this is at. And look at a bicellular microspore, which would typically be 2 days later. And it's totally a different situation. Almost everything's monoallelic. You can start to draw the haplotype without anything sophisticated. Just looking by eye, you can pick it out. So there was this huge shift about 10 days after meiosis, right around the first pollen mitosis. Now, I was also interested, could there be some categories of genes, maybe even if they're rare, that would be expression of the haploid genome early? Are there going to be categories even rare that persist as biallelic transcripts later? And to look at that, I needed a way to distinguish on a gene by gene basis haploid expression from other ways you could get monoallelic gene expression. And again, it's actually not that difficult to separate haploid from everything else. Here's an example of one gene that was consistently from B73 in pre-meiotic PMCs. So some of the genes you see out here can actually show the strong allelic type bias. But look, if I take a random gene from a pollen grain, it's again consistently from one allele, but it's half the time that allele is going to be B73, half it's going to be A188. And so it's just a simple way to separate these. What I did was calculate for each individual pollen grain or pollen precursor, what percent of all transcripts come from the B73 allele and which percent come from the most abundant allele. I excluded any precursors in a stage that had fewer than 10 transcripts, just because you start to add a lot of noise when you have really low counts. So in this case, these 6 precursors would have been average. I find about a third come from B73, but almost all are from the most abundant allele. And that way you can start to see how much this looks like haploid expression or not. So here's a graph of these two quantities for all genes looking at all meiotic stage cells. So a single dot here could be like this gene here would contribute to one dot where it'd be heavy amount B73, heavy amount monoallelic, and the x-axis is giving that first quantity, how much is B73. This one's giving which percent's monoallelic. So you can see there are some that are coming primarily from one allele, but it creates a straight line here. What that means is in every cell, it was the same allele that was more common. So these are measuring the same thing. Now here's what a pollen grain looks like. It's a very different story. Almost everything is monoallelic, but it's coming from a mix of both alleles when you could compare between pollen grains. So with this, I could better identify, haploid expression from other forms of monoallelic expression. You can see there are some cases that are consistently from one allele in B73. I've not looked at all of these in heavy detail. It's a small portion. A lot of those, I think, are presence-absence variants. It's things where you only see from one allele or the other for many of them. But I think there are probably some other cases too. But here's what happens when I look at the post-meiotic stages before pollen mitosis I. So here's tetrad, and this is the tetrad's been dissected. So these are individual cells of the tetrad, so I can actually identify haploid expression if there is any. Here are the uninucleate microspores, so the single cell stage. Here are bicellular microspores, and you can see, even looking across all genes, there's nothing that I'm detecting that would hold up to any statistical tests that is expressed from the haploid genome before pollen mitosis I, and then it's pretty much everything is expressed from the haploid genome after pollen mitosis I. Two things I want to mention here. There is genetic data where we do know of certain things that cause phenotypes before pollen mitosis I. A really great resource for this is Ed Coe in the early 90s looked at a lot of hemizygous lines where you're basically missing a chromosome arm. And he looked at 17 of the 20 maize chromosome arms. All of those result in 50% dead pollen. 7 of them have phenotypes that accumulate right at pollen mitosis I, which becomes a little bit easier to explain when you think about all the genes coming on there. But 10 of them actually have phenotypes earlier. So there has to be some expression earlier. There's just nothing, it has to be a really rare class. You know, if I do order of magnitude and assume that genes required prior to PM1 are randomly distributed across the chromosomes, the genetic data would lead me to about 13 or 14 genes are required from the haploid genome before this mitosis. And if I assume that strongly or weakly expressed genes aren't more likely to be required, I could put a upper bounds around 40 to 45 genes that I could have missed even if they were expressed from the haploid genome early. So I do think there is some expression early, but it seems to be a very rare exception with this huge trend towards the pollen genome being activated very sharply around pollen mitosis I. The other thing I want to mention is just due to sampling, my time resolution is probably about a day. So I can't tell you, is this just after, just before pollen mitosis I, if it's perfectly correlated with a certain stage of it. I didn't capture any intermediates here. So it's something relatively sharp, but that still puts the time frame probably within a day of the 28-day time course I had. And we're looking to get more data focused around pollen mitosis I so we can better capture intermediates, link it to the cell division itself. But right now, I just know it's near that cell division. So how is this regulated? And you know, to give you an idea, here's one of those graphs I showed earlier for C. elegans maternal to zygote transition. And in species where that's been studied better, mostly in animals in particular, there's usually some coordination where you have regulated period of degradation of maternal products that can actually occur a little bit earlier with different waves of zygotic genome expression. So really, you know, as a first pass, I want to understand how much of this shift is due to degrading those prior pre-meiotic transcripts versus new transcription. And that can be a tough thing to analyze carefully from gene expression data, but there's a lot of features in my data set that allow me to address this. First of all, if, you know, say we have a pool of transcripts, these red ones are representing some gene, gene A. You know, typically we might see, hey, gene A is upregulated, and maybe this is a hint that there could be more transcription going here. But there's a couple models that can often explain that same data. For instance, often when we do transcription, it's RPKM, FPKM, TPM, something per million, transcripts per million. And so with those data, we can't distinguish a model where it's just, what if the total number of transcripts are going down, and this is just that one transcript that's persisting? So you either have to assume that the total number of transcripts don't change, or you need some ability to measure the absolute abundance. And that's a really nice thing about this sort of data. These pollen precursors are a closed system. After meiosis is over, there's no plasmodesmata, there's no interchange of RNA. So I'm looking at the full, you know, the subset I can make libraries from of RNA from a haploid pollen precursor. So I can actually estimate absolute transcript abundance. Based on what we know for numbers of pollen genes, I probably get about 20% of all transcripts I actually detect, but that number seems to be plausibly consistent between stages. And the absolute amount of transcript abundance is not static during pollen development, and so that assumption would not hold very well here. Here I'm showing these long stages again, and so this is meiosis, and you can see a slight decline. At the end of this is partly due to the meiotic cell divisions. You're going to half the number of transcripts per cell with each cytokinesis, but there's, in total between late microspores and before meiosis, there's about a 16-fold drop in RNA levels. So a fourth of that is cell division, and a fourth is degradation being more dominating over new transcription. But then look what happens at Pollen Mitosis I. There's this big jump and about an 8-fold increase in new transcripts right when I see the things shift to the haploid genome. And this is true for most genes here. So what I'm plotting, this is the log 2 fold change in the transcripts per pollen precursor, where I'm comparing bicellular microspores, so after the mitosis, to late unicellular microspores, so just before that. And you can see most genes here are changing over 2-fold in terms of absolute transcripts per pollen precursor here. There's a small number that seem to be degraded, but it really does look like new transcription is dominating that shift to haploid expression. Other pieces of my data that make it easier to distinguish new transcription from degradation is any new transcripts after meiosis can only come from one of the 2 alleles. And if I look at these genes going up, consistently, whichever allele is most abundant skyrockets. But there still is detectable expression of the other allele, and it's often pretty similar to what I found in the earlier stage. So I think that shift to haploid expression is really just huge upregulation and activation of the haploid genome, and that swamps out whatever transcripts are hanging around at the end of the microspore stage. So, want to compare a couple things about this sporophyte to gametophyte transition with the maternal to zygote transition, because they're actually surprisingly different compared to what my expectations were when I began this. One, they both represent a shift between expression from the parent to the offspring, and kind of shows a greater parental investment in the offspring, right? Here we're putting a lot of transcripts into these microspores to power the first 10 days of haploid development. So the sporophyte is contributing not only externally, but also internally during the beginning of haploid life. But there's some really big timing differences. The maternal to zygote transition ranges from about 3 to 30 hours if you look at relatively quick or long animals. And sporophyte to gametophyte, we only have data in maize now, but we're talking like 10, 11, 12 days. So it's a much longer time period with those parental transcripts dominating the gene expression. In animals, the maternal zygote transition occurs in waves, while the SGT seems pretty sudden. Now, again, my time resolution is about a day. I'm sure as I get more data, I'll be able to find some intermediates. But relative to how long it waits, that's a pretty sudden shift to have a 10-day lag, and then it's all done within about 24 hours. And it primarily seems to be driven by new transcription. So regulated degradation's a little bit of a role, but it's mostly just huge activation of the haploid genome competing out what was left over. So a couple ideas for why we might delay, why this transition might wait until Pollen Mitosis I. And my leading 2 hypotheses, probably my favorite, is that this has something to do with protecting the germline. That first mitosis is when you set up the soma, the vegetative cell, compared to the germline, the generative cell, for the gametophyte. And so by keeping the chromosomes condensed in the haploid microspore until you have the vegetative cell, which is probably where a lot of the new transcription and metabolic activity is coming from, it helps prevent things like transcription coupled damage, but even more might have to do with access to transposons. In animals where maternal zygote transition has been looked at a lot, it's very common to see whatever lineage is going to the germline waits a lot longer until the offspring genome is activated compared to the rest of the cells and the organism. And for transposons, when activity is happening in a somatic cell, evolutionarily, that transposon is aligned with the host. If the transposon takes the ship down with it, it does nothing for that transposon. So it's really only when you have cells or DNA material that can go on to the next generation, does the transposon benefit from replicating and duplicating even at a competing negative consequence on the host? So by trying to lock things down until you have the soma and then other pathways that might help try to prevent transposons from hopping from the vegetative nucleus to the germline, this might be a way of really limiting how long transposons can be active where they're in contest with the host in terms of their goals. Another idea is that this is just history. It's a legacy of germination. You know, presumably the last common ancestor, like earlier common ancestors of plants, spores would have been released in the environment, and germination was an environmentally triggered event that would cause that cell division and growth into the plant. And a lot of ferns, for instance, germination is accompanied by that first mitosis. So in angiosperms, there's no more spore release environmental germination. It's become a developmentally programmed event. But maybe this whole genome activating used to be what germination was, and as that got reduced and captured inside the sporophyte, we've still conserved that timing compared to what happened before. And like many things in biology, it can always be all of the above. So last thing I want to leave on this question is just a little bit of what portion of the genome is actually expressed from the haploid genome. So here's two pie charts where I'm looking at sporophyte versus gametophyte. I use my same library prep on just whole seedlings and define sporophyte as anything expressed at a certain level in either whole seedlings or in meiotic cells before the first meiotic division. And then gametophyte is everything tetrad and onward. And if I just say, what do I detect at least once? It's about 87% are in both. If I require a little bit more stringent threshold of 100 transcripts per million, it's about 54%. You know, these quantifications are always going to be sensitive to your threshold of detection, but this fits kind of what's been seen before. Typically, when it's been looked at, we're talking 60 to 80% of genes are probably present in both phases in the majority of plant species that have been looked at. Now, what I can add to this, though, is I can look at, well, which portion of these are actually expressed after pollen mitosis I? Because if they're absent at that stage, you know, the prediction would be they're not really expressed from the haploid genome. These are lingering on from meiosis. And so this does cut down the number a little bit, but we're still talking a huge portion of the genome that might be subject to selection in the haploid phase of the life cycle. Now, I looked at these gene categories a little bit more just to see some early evidence of, is there any more or less selection on genes expressed at different stages in pollen development. And here what I'm plotting is DNDS. So this is the number of non-synonymous substitutions per non-synonymous site to synonymous substitutions per synonymous site. So basically, are there a lower value means there are fewer substitutions in things that would change proteins compared to substitutions that aren't. So this right here is just everything that I don't detect after meiosis is over, so not present in the gametophyte. If I look at genes that are expressed, at least detected, present after meiosis, but absent after pollen mitosis I, it looks pretty much the same in terms of DNDS as the genomic background. And you know, if I draw up my time course, what I essentially mean is it's present in these stages, but not later. Now, if I look at genes that are present in bicellular microspores or pollen, there's a pretty clear decrease in DNDS. So less non-synonymous substitutions are permitted genome-wide. And so this is some evidence of purifying selection occurring in the haploid phase. And one thing that strikes me, this category here, this is after pollen mitosis I, but absent in mature pollen. And it looks the same as if I look at the genes present in pollen. So that suggests that you might be having some real selection, at least purifying selection, before even pollen shed, because there's a lot of metabolic activity that occurs getting pollen ready to be shed in germination between pollen mitosis I and mature pollen. So, you know, some conclusions: diploid-derived transcripts persist long in the haploid phase. There's a really rapid conversion to monoallelic expression around pollen mitosis I. And a couple hypotheses for why that might be, but at the moment we don't have any clear evidence to just one or the other. I think I'm going to try to go through quick so we have time for questions. But I did want to end on a little bit of some of our work to take the abundance and some of the unique features for selection on the haploid phase and use this for genetics. So when we think about the sporophyte and most of our breeding, genetic screens, everything, you know, there's a limit to how many plants we can have in an acre. Compared to the gametophyte, we have 10 million pollen grains per plant. So if we can study anything we could study in pollen, we could potentially use more microbial-like genetics in terms of the scale and the ability to select directly on pollen. And so what we're doing is taking an approach that was originally developed in baker's yeast and has also been used in some funky haploid human cancer cell lines and Chlamydomonas algae. And the basic idea is a way to do a pooled loss of function insertional screen. So I'll briefly explain how this has worked for Chlamydomonas. An early screen for this was looking for genes involved in photosynthesis. But Clammy, they've made many different insertions in random locations in the chromosomes. This would be equivalent to the Arabidopsis tDNA library, but they have lots and lots of plates of these with many different lines. So that part's just like Arabidopsis or maize mutator populations. But the big difference is they can mix all of these lines together. And then what they did is they took that, split it, and in half of it, they grew for photosynthetic growth. So it's under light. You need the light and photosynthesis to get your carbon, or they grew it for heterotrophic growth. They providing carbon in the media. There's no light there. And then they sequenced to figure out all the different insertions in this population. Are there any where the abundance is different between these two? And what they were really looking for would be cases like this blue representing a specific insertion in the genome where it's being outcompeted when you're requiring photosynthesis, but it's doing fine when you're not. Here's a graph showing this. Each of these dots is a different insertion in this library. So you can get a sense of the number of insertions they were able to screen in just a vat. And what they're showing here is the read counts in the photosynthetic growth versus heterotrophic. And so they could find cases like this where they observe it a lot in the control, but not the test population. And they actually discovered some new genes and required for photosynthesis in algae. You know, and this is a type of screen you can really get towards saturation. There's a lot of different ways you can think about it. And so we're trying to bring this to multicellular plants in terms of pollen. This, as I mentioned, it's been used in a lot of different circumstances, most often in baker's yeast. But the basic format is you take a haploid organism. This way you can put the insertion in and you can immediately get a phenotype. So insertional mutagenesis. It's been done with random insertions and transformation, transposons, anything that you can track later by sequencing. Then you screen your select for your phenotype of interest, and you quantify the insertion abundance in that test versus a control population using sequencing. So one way, the main thing that we're working on first to test this is a classic maize transposon that's really powerful for insertional mutagenesis, which is called mutator. Classic phenotype is you can actually see the transposon remove itself once it's been in. So the speckles on these kernels are because this transposon was in an anthocyanin color gene and it's popped out. But it also has a really high insertion rate. And mu active lines you can have on the order of 50 new insertions per pollen grain, and it's trackable by sequencing. So, you know, for example, one way that this could be applied, you can have a mu active line subjected to heat stress or not, and you could look for insertions such as one in this you know, represented by this blue population that are depleted in the heat stress, and therefore this gene might be required, or this part of the genome might be required to respond to heat stress. And so we've kind of adapted one of several different approaches to sequence mutator insertions, and we made a couple changes to the biochemistry and the analysis to make it more quantitative. This is work done by a technician, Grant Freeman, in the lab. But just to show you that we have a quantitative way to look at these insertions, mutator has a terminal inverted repeat, so it's a reverse complementary region on each end of the transposon, and we basically shear the DNA and sequence in to see where this boundary is. What I'm showing here are insertions that we detected in a single hole punch size of a seedling leaf. And this is the number of times an insertion was detected in this direction compared to the corresponding times it was detected in the reverse direction. So these would be two independent DNA molecules for amplification, but because each transposon we can sequence out of each side, I'm just showing you that I sequence out of one side a similar amount to how I sequence out of the other. So you can see a real nice correlation here. These really abundant insertions where you kind of see a cluster. We just have data back with more plants in the family and we can confirm this, but I'm pretty sure most of these will be ones we see segregating in the family. These were the inherited mutations, so they're going to be present in almost all the cells of the population. But then we can see insertions across several log orders of magnitude that we can consistently measure here. So we have a good way to track insertions, even rare new ones in a population of DNA. And, you know, I mentioned heat stress as kind of a motivating example, but, you know, one thing is thinking of this kind of screen, you know, I'd encourage you to think pretty broadly about what we could actually model in pollen. You know, every aspect of cell biology, metabolism, and the plant, almost every, has some role in pollen. A lot of things about developmental biology. You have an asymmetric cell division, establishment of the germline, all kinds of gene regulation. A lot of the haploid inducers are gametophytic effects, you know, responses to stress. So there's a lot of things, even if your end goal isn't pollen, that we could potentially look at in pollen. And the two things that we're actually working with to start aren't heat stress, but one is we're looking similar to what I showed you with starch biosynthesis. So basically we take a mu active line, we're staining with iodine, super old school techniques, and then we're sorting the pollen so we can see what insertions are leading to unstained pollen grains compared to the stained ones here. Now we can follow up with this, and the prediction would be, because a lot of the starch biosynthesis is also used elsewhere in the plant, that we might be able to identify some hopefully new genes in starch biosynthesis that play a role at multiple different life cycle stages. So it's kind of a nice test case of these kind of screens. I'm very interested in understanding different aspects of pollen development and gametophyte development. Currently, we've not been able to really access essential genes in plants, because if you can't transmit anything, even in a heterozygote between the male or female gametophyte, almost any way we've screened for stuff, you're going to lose any of those mutations. And this kind of screen, we can actually look at what happens immediately after the mutation was made in the sporophyte. And, you know, one example, we've come up with simpler ways to do this by now, but we could, the initial idea was actually to isolate pollen precursors at different stages and look for insertions that drop out going from one stage to the next. So we can find time dependent which genes are required to get to this stage or to this stage. And so looking at this for developmental genes, you know, but the big thing is with pollen, I think this is really an opportunity now with some of the power of what we can do with genomics and with sequencing to start to use these very small organisms to think about a lot of cool things with plant genetics. So with that, I'd like to acknowledge my lab. Jenny Walbot's was a great mentor in my postdoc. Chen and Lu shared the A1A genome sequence ahead of publication, which was very helpful for this particular project. And I'll take your questions.
Thank you for the very nice presentation. Are there questions?
Yeah.
Do you have you looked at any changes in the DNA, like methylation changes, right at that, what I call actually microspore mitosis, but you call pollen mitosis the first one?
Yeah, yeah.
Do you see differences in methylation or something that would suggest that this is the trigger to get the haploid machinery going?
I haven't looked. It's been analyzed in Arabidopsis, and there's I think it's recently published in maize. I need to dig into this data a bit more. There are some pretty big methylation changes, especially near like the centromeres, But I want to look at that a bit more to see how that makes sense. We're thinking about going for ataxy because the hypothesis there would be a pretty big genome-wide opening here. There is a lot of available data that's been looked at in the context of transposon activity and in other types of biology that we need to take a much deeper look at. But I haven't gone very thoroughly into it yet.
It'd be interesting to see how that pattern goes, because
Yeah.
then when you think about the sperm nuclei being formed, and then the idea there is, oh, it gets all packed and methylated again.
Yeah.
So it'd be interesting to see what that might look like.
Yeah, so we're thinking a little bit about some of those sorts of approaches. I'm not sure if we'll go beyond ATAC. Part of the issue is I'd be shocked if we don't see some pretty big changes at this time, but I don't totally know what I do next with that information, right? Because if the whole genome's changing, we kind of are already seeing that by one way. It helps confirm that. And so I'm really prioritizing things that might help us get closer to what happens first. And that's where even the exact technique I've used, but taking a lot more pollen precursors, now isolating that stage, just because there's got to be something that's earlier than another. We were considering some experiments of using kind of sledgehammer approaches, like you block the cell division, you block the ribosome, you really hit something that's going to shake things up, and then look just 2 hours later with the hope that maybe we can trap some intermediates, because maybe those first transcripts can turn on, but now nothing can happen downstream if you block the ribosome. So we might play with some of those to narrow down. If I look at transcription factors, chromatin genes, stuff like this, the numbers that go up at this stage are enormous. And it's a little bit of a challenge because the ideal gene probably isn't a gametophytic one, but it's one that was on before this all happened. Yeah.
You showed that for one of the VM and pollen, there's 5% and 3.5% for the weakly monoallelic.
Yeah.
I'm sure you have played with the threshold. What was the, is there any way to remove that completely, or you think that is actually happening biologically? Through the sequence reads or the standard or threshold you're using?
So this is, you can see how some of these are dipping or like not quite on the line here. Like they're a little bit below 100% and stuff like that. This is pretty reproducible in the bicellular stage. And if you go to pollen, I'm not sure if I have one. Here we go. You see it's a lot sharper. And I do think there still is some persistence of transcripts from before. I think that's a biologically meaningful thing. But you can see how much those are swamped out by the new transcripts being made. In terms of the thresholds and stuff like that, it gets tricky because there's a lot of technical things that can also lead to these differences. The reason I picked the 80% threshold is that was actually, I think 80% to 95% seemed like the reasonable range to consider. And I went lower because that's less stringent for calling something monoallelic, but it's more stringent for calling it biallelic. And the big surprise here to me wasn't that the bicellular stage had a lot of genes expressed from the haploid genome, but that the stage before that was still biallelic. And so I wanted a threshold where you could see really that most of the genes really have a clear portion coming from both alleles. So that's why the one I chose for a lot of my figures and presentation is stringent for biallelic, but less for monoallelic.
Yeah, I was wondering if you have any way to tell if these are expressed in the sperm cell, vegetative nucleus, or both, either in your own data or.
Yeah, so there is data, including in maize, with sperm versus vegetative cells and stuff. I've not gotten around to going for that, partly because I would be absolutely shocked if it's not almost all vegetative cell. So I'm kind of, maybe I'm assuming that too much, but there is a lot more expression from the vegetative cell. And so I kind of would expect any comparisons I make with that, it's going to be almost all vegetative cell. And maybe you could use this to look at some newly expressed things from the generative cell or something like that. But the thing is, it's so much all or none for things being upregulated. So I don't know that my data would shed new insight into what's turning on in the two cell types. And I think based on plenty of prior data, I really do think this is mostly vegetative cell I'm looking at. So, I mean, I do, my hypothesis would be that this transition is actually just before pollen mitosis I, not after. And there's a couple reasons for that. For one, there are mutants, especially in Arabidopsis, that block pollen mitosis I that are gametophytic, suggesting, you know, a number of genes that clearly have to come from the haploid genome prior to pollen mitosis I. There are experiments, both genetic or using chemicals, where you block pollen mitosis I in a few species, and by most accounts, the pollen grain develops kind of normally. It continues to fill with starch. You can actually extend a pollen tube. That pollen tube can seek out the ovule, even though there's no germline to deliver. So I kind of think a lot of what's happening in the vegetative cell is really dominating, and doesn't require the mitosis itself to continue. Now, is this genome activation required for the mitosis? There's not any data that help give me any expectations for that. But my money would be this transition occurring just prior to pollen mitosis I. Kind of conveniently for me, a huge number of pollen promoters that are used turn on right around pollen mitosis I, which makes sense in retrospect. But it means it's not super hard to find good promoters for reporter genes for this transition.
You screened every stage of micro-sporophyte-to-gametophyte and mutagenesis. Are these different stages at the same times or different times? Now, is it a destructive method of extracting these ANC? Or what's the difference?
It's a destructive method. I mean, you could very easily harvest a spikelet, collect material, then continue to harvest from the same plant. So there would be ways to do this all from one plant, but that would make things a lot more difficult. And so this is destructive method, multiple plants. It's definitely destructive on the per cell or pollen grain basis. Typically from one plant, you get about a week's worth of time course across the tassel, depending on where you are. And from one plant, usually I would get multiple different stages collected. And I made sure every stage I had at least two plants. And in the beginning, the stages were just based on anther length. And then it was subsequently staged by microscopy to know what stage I'm at there. But I covered each stage with multiple plants, and each plant contributed to several. I didn't see any clear differences from plant to plant. Like, you don't get clustering based on plants beyond what the stages that you happen to isolate would tell you. So
I'm interested in the selection that you mentioned at the beginning between the gametophytes at the pollen level. This race kind of.
Yeah.
But do you think that is only to get rid of junk ultimately, or do you think there's also kind of the more vigorous gametophytes, or the ones that make the pollen tube the fastest? They contribute to also making the diploid population better.
Yeah. So Mulcahy, who had that kind of really cool hypothesis that I presented, a lot of his work was actually in maize, and some of the things he showed is that if you had two different lines and one has a faster pollen tube, it would correlate with having a faster growing seedling. And you could compete those and have the faster pollen tubes win a lot and have faster growing seedlings after that. And so he showed a lot of things where you could correlate some level of seedling vigor to pollen tube growth.
So would it mean if you leave a population alone and outcross, it should just become more vigorous or better? Because always the gametophyte.
Yes, that would create a selection for some level of metabolic vigor, but a lot of what we work with has had a long time of evolution to optimize for many things. So, I think in many cases, purifying selection is a big part. You have new mutations arise, and this is an easy way to remove ones that are bad. But it certainly increases positive selection also, and can lead to competition for faster growing. I most of the time when I talk to geneticists about this, the thought's always about loss of function genes purifying selection. and I talk to evolutionary biologists, and it's always been like, well, in what cases pollen grain in competition has different needs than what will happen, like antagonistic selection. And I'm sure some of that exists, but I don't know of any examples. My guess would be the most common thing is purifying selection, a little bit of selection for fitness. But
I was also wondering if there would be a difference to be expected between insect pollination, you said
Yeah.
all start at the same time, versus wind pollination, where they are coming one after the other, and it's maybe not as.
So Sally Otto's a great kind of evolutionary theorist, and she's thought a lot about some of these questions, and might there be actually evolution of more or less haploid selection under different circumstances, and outbreeding versus inbreeding populations, insect, wind pollinated. There's a lot of hypotheses you could raise. And when I started this project, I was fully expecting, one, it would be waves. It's not going to be all at once. I thought it was going to be a whole lot earlier than it was. And my expectation moving to other species was that it would be quite different when I look at other species. The maternal to zygote transition in animals can be in the zygote or hundreds of cells later. It's not a conserved point of time. I'm actually now, like, we're looking at some other species now to get a sense, is this conserved? And my leading hypothesis is now actually it will be. Part of the reason is there's a lot of data that all hold together with Pollen Mitosis I being a pretty big time of genome activation, where in retrospect, it's like, oh, that's why, of course, that promoter reporter would turn on at that time, or this would happen. And, but it's also just, it's such a long delay that makes this a lot more striking, how all or none it is. And the fact that it does correlate with when you are separating germline and soma, I'm wondering if it might be a more conserved thing, at least within angiosperms, and I don't know how far back. And so far no plans to look at the female gametophyte yet, but we might finally roll up our sleeves to go for that challenge at some point. Yeah. Yep.
Could this transcriptome be different from other crosses?
It totally could, and this is the only line that I have any info on right now, but that's one of the things we're wanting to get at next is what's the time scale of conservation for this, right? Because on one extreme, every inbred line could be totally different, but on another extreme, if we look at a fern spore, it's going to be that first mitosis around when the genome gets activated, and anything we look at, we're going to reproducibly see that. So we're starting with diploid relative of tobacco and Arabidopsis, partly because they're useful models to us or others, and probably rice or sorghum. And we'll see how conserved they are. If they all seem to be all or none around Pollen Mitosis I, we might go a little bit further back in evolutionary history to see how far it goes. If they're different, then it partly depends on, are we seeing something suggesting monocot, eudicot, or that all of them are totally different from each other. And it definitely changes the story a bit. It's cool if it's super conserved. It's cool if you might have wind pollinated and insect pollinated things having, selection for different timing of haploid genome activation.
Okay,
cool.
No more questions. Thanks again.
Yeah, thanks for having me.